Everyone, my name is Grace Peng from the National Institute of Biomedical Imaging and Bioengineering at the National Institutes of Health, and I would like to welcome everyone to the Interagency Modeling and Analysis Group Multiscale Modeling Consortium webinar series. I'm pleased to introduce the speaker for today is Dr. Tom Royston from the University of Illinois in Chicago. He is currently the professor and head of the Department of Bioengineering. He's also a professor in the Department of Mechanical Engineering, and he's been directing the UIC Acoustics and Vibrations Laboratory since 1995. I'm pleased to say that Dr. Royston is also an NIH NIVIB NAGI New Investigator Awardee from my own institute. And so today he will be talking about his Audible Human Project, which is a project supported by the NIVIB, and he will talk about his computational simulation model of how sound and vibration are generated and travel throughout the body. His talk is entitled The Audible Human Project, Seeing Touching Sound in the Body. Thank you, Tom.
Okay, thank you very much, Grace, and thanks for everyone tuning in. First of all, I'd also like to acknowledge my editors on this long-time endeavor, Dr. Nancy, Richard Sandler, and Bob Buck, who, when we all started working together, they were all at Rush, which across the street from UIC. Bob is still at Rush, but Hanson and Richard have moved on to warmer climates down in Florida. This work, they're absolutely critical to this ongoing project. So, Audible Human Project. Great. Let's see here. It's frozen. Okay. 1 second here. Okay. So what is the Audible Human Project? So the name I came up with basically as kind of a play or a complement to the Visible Human Project, which was developed by the National Library of Medicine. A lot of us use the images that are available there for research and aid purposes. The big picture, as Grace mentioned, is with the Audible Human Project, the endeavor is to develop a comprehensive computation model of sound and vibration in the body. So it's a fairly broad goal. Over the past 5 years, where we've been funded by NIH, our focus has really been on pulmonary acoustics, and that's what I'll show today is related to pulmonary acoustics. So ultimately, the deliverables we want to provide are an externally validated computer simulation model of vibration in the pulmonary system. Along the way, we are developing some novel modeling and simulation approaches, and we're also doing a lot of experimental validation, which involves developing some innovative imaging and experimental methods. Okay, why? Why would we do this? Why should we be funded for this? I'll offer several reasons. One, iconic image or symbol for the medical doctor, the physician, the stethoscope. Something that's qualitative, arguably subjective, single point, very skill dependent. If we have a better understanding and ability to simulate, for example, respiratory sounds in the body, and how they're measured, we might make this type of device more quantitative, more objective. If we think of array measurements, we can start to get more spatial, temporal information, more automated. There's a little interest, there's always been interest, but it seems like there's a lot of emphasis now more on mobile health, home monitoring, diagnosis and monitoring. So be bringing the stethoscope or other types of similar devices into play, and it would be nice if we could improve what we can acquire with them if we have a better quantitative understanding of the respiratory sounds and other types of sounds that are created. Another interest, why develop the Audible Human Project, which is linked to a lot of my other related research, is the area of dynamic elastography. which basically there are different ways of capturing images of vibration traveling through the body, be via ultrasound or phase contrast MRI or certain optical methods. A lot of these approaches tend to focus on using a single frequency. These days are more focused on, say, the macro scale, looking, for example, at a large soft organ like the liver, and getting an image and then looking at the shear waves that are in the organ, and estimating for in this case, staging liver fibrosis. Now, a better model of how sound and vibration are going to travel through the different parts of the anatomy is that we could start to look at broader ranges of frequencies, look at things at different scales, look at more complex anatomy. In a couple of examples here in the lower right, I'll talk a little bit more later. This is one of our simulations of sending sound into the airways of the pig lung, and then calculating what is the sound field in the airway structure as well then as the sound and vibration that gets committed into the surrounding parenchymal tissue. On the lower left here, these are actually a couple of experimental images. We're doing MR elastography on a mouse, which has a heart that is basically the left ventricle there is highlighted in the middle figure, has a of about 1 centimeter, is beating 500 times a minute. The mouse is breathing 100 times a minute. And somehow we're trying to capture images of where we're applying vibration on the sternum of the mouse using a stick source and trying to capture and how those waves are propagating through the torso of the mouse. And we're gating with the cardiac cycle, we're gating with the respiratory cycle, and we're trying to see if we can identify changes in stiffness of the ventricle that goes throughout the cardiac cycle. So I'm showing here some snapshots, animations of the wave field that we were able to experimentally capture in diastole and systole. And I guess the point I can bring this up is that this is a lot more complicated to try to understand what's going on here as compared to, for example, the liver, where it was clear, obvious to see these shear lengths. Here it gets much more complicated structurally. And we have a finite element model of this mouse that relates this experiment, and we use that to help us try to optimize the acoustic source that we could properly vibrate the ventricle of the mouse without harming the mouse. And it's quite a challenge in terms of the proper arrangement, the proper frequencies, etc. And so there's another need for a model. But without the model, in terms of interpreting the data, the best we could do is identify a small portion of the left ventricle showed in the lower figure there, where we had confidence in our measurements throughout the entire cardiac cycle. We can construct the graphs on the right here. The upper one, I'll just focus on a little bit. We're able to track how the stiffness of the LV is changing through cardiac cycle, whether we're going from diastole into systole. So we're kind of giving explanations for why do we want to have an ability to model sound and vibration. So the first two focused on diagnostics. A more fundamental argument, I would say, has to do with understanding dynamic mechanobiology. For example, the effects of dynamic forces on cell tissue remodeling. And then what the effect of those changes in the cell and tissue structure have on the acoustics measured at, let's say, at the organ level. So this is work I have done related things in the past. We've done studies looking at fracture healing using pulsed ultrasound. And the thing that still to this day kind of puzzles me with that is pulsed ultrasound has both ultrasonic and double frequency, low frequency vibratory attributes to it, and never was quite sure what was it that was affecting this angiogenesis and tissue regeneration. Maybe a combination of both. Also been involved in studies where we've looked at vibration in the vascular system. And, there's been a lot of research on stress, shear stress and its effect on vascular remodeling, but there's also a lot of vibration that goes on anytime you have traditional or turbulent flow. We did a number of studies trying to correlate these things through some computational simulations and experimentally. We also looked at kind of studies of the incidence of, for example, acoustic stimuli, as well as the shear stress effects on cell morphology and gene expression. These are a few figures taken from some of these articles. This is back about 10 years, where we're trying to model the flow field, the turbulence, and we're also trying to take measurements. And it's a very complicated problem, and I think it would benefit from a better understanding and ability to computationally model, not in this case, just not the flow field, but also the structural dynamics and in the fluid structure interaction. Finally, the fourth reason why we want to develop the Audible Human Project. Something I'll want to talk about a little bit today is some of the executable free software programs we're developing for interpreting acoustic measurements, elastography measurements, as well as ways of viewing them. And I'll have a bit of a demo of the ahp.exe later. So we're looking forward. We're interested in taking this to the next level in terms of visualization. And here I'm tapping on the expertise of one of my colleagues here, Christian Luciano, where we try to integrate some of this visualization into a virtual augmented reality environment. And because we have properties, for example, from elastography, we can have a haptic aspect to that as well. And I'll have a little bit of a illustration of that later. Although on a two-dimensional flat screen here, you're always going to lose something relative to being in truly seeing kind of a 3D rendering of it. Okay, so let me focus a little bit more now on the lungs and show some of the work we've done in developing this project. The lungs are very interesting acoustically. A lot of different conditions result in changes in mechanical properties that lead to acoustic changes. So one of the things we've studied for a long time, particularly with Drs. Balk and so physicians on our team, have been pneumothorax and finding quick acoustic ways to confirm pneumothorax. And so some of our early simulations have focused on that. That's kind of a big change in the geometry of the lung. We can look at other types of pulmonary diseases, whether they be airway focused, such as various obstructive lung diseases that are going to alter the structure of the airways and the alveoli. And we also look at other restrictive diseases that are kind of altering the mechanical structure of the interstitial tissue. Within all these things are going to change the acoustics, the mechanical dynamics of the lung structure. We also, of course, look at cancer. Tumors, of course, are changes in stiffness, changes in density that change the acoustic field. So what are the components that go into developing the Audible Human Project? Well, we've spent a lot of effort on understanding viscoelasticity, and this has included also coming up with different types of ways of measuring it. Here I'm just showing a few examples of optical methods if we have a line of sight using a stroboscope top center or using laser vibrometry, some of these other figures. And typically what I have here in the lower right is the type of plot we can get where we can, in this case, we're adding a measure of the shear storage modulus and the shear loss modulus, which both be highly frequency dependent. We can also do this internally using, for example, resonance elastography. And our interest here has been in seeing over how many scales, over what frequency range, in this case, can by MR elastography. And here, this is a exotic here from taking EchoFlex, a tissue phantom, and seeing if we could create a shear wave field over a very, very frequency range. And then we were studying different types of models of viscoelasticity. So up here are some of the conventional models. Bottom left here, what we found works most of the time, that consistently works the best, is something we call a spring pot. This is using fractional calculus. So the spring pot here is kind of, for those unfamiliar with fractional models of viscoelasticity, is kind of it's a mix of springs and dampers, and actually asymptotically, if you take this fractal-like arrangement of spring dampers, asymptotically, you'll arrive at the spring pot, which is characterized by a fractional derivative, which in the frequency domain is very, very simple to deal with, and we are typically working in the frequency domain. Generally, when we have linear system acoustics here, it's easy to go to the frequency domain and do things in the frequency domain. We found this typically works the best, and it may surprise it. We find this with both biological tissue as well as with complex molecule polymers like the EcoFlex, which I had shown in the previous slide, and which I'm showing some results for here. We'll have mechanical forces operating over multiple length scales. So this was from a paper we had. We did this broad range study from 200 hertz. We've actually now gone up to 16 kilohertz, looking at EcoFlex and looking at different models of viscoelasticity. Basically, find that we're calling this fractional void, which is a arrangement of a spring pot and a spring, consistently will give you the best fit to the data with the least number of parameters. Basically, we just have two parameters here that define the spring pot. Okay, so another component that we're very interested in modeling is the lung structure itself acoustically. They're porous. For the past several decades, the approach to modeling the lungs has been what we call a closed cell approach, where we'll take the stiffness of the tissue, the stiffness of the air, and in a reciprocal way, we combine them to get an effective bulk modulus and take basically the average density. And this gives us the phase speed of compression waves, C sub P. And it's interesting is that we'll have that basically the compliance of the air kind of short circuits matters, but we still have the density dominated by the tissue. Predictions of phase speed on the order of 20 to 30 meters per second, even though the speed of compression waves in air is on the order of 350 meters per second, and in soft tissue is on the order of 1,500 meters per second. For evaluation, we'll have traditionally there's been an approach of treating it as what we call like a bubble swarm, where you'll have scattering, and so bubbles being, in this case, the alveoli, thinking of them in terms of closed cells that are scattering the sound field, and some kind of thermal dissipation. So that's been used for years. Recently, we've been looking at applying BO theory to the lungs. And, this involves a set of coupled equations linking the mechanics to the flow. Not a closed cell formulation. We're allowing for flow through the channels to a limited degree. And the parameters in the BO theory, even though this is macroscopically describing how sound propagates through the lungs, these parameters are based on microscopic characteristics of the lung, such as the dimensions, for example, of the alveoli. So we've done a lot of work with this model, and basically, as my former Ph.D. students, Lejun Dai, we are doing an experimental study and show that this model actually works better at predicting the compression wave speed than the traditional effective medium model. So we incorporate this into our simulations. It does a much better job of predicting the attenuation. What I'm showing here is experimental results at two different inflation pressures on a excised pig lung, which we used for this validation study. So another part of modeling the acoustics of the airways is the airway structure itself. This is a diagram taken, I believe it might have been from Fredberg, one of Fredberg's articles. I reference it here, where he and others, Horsfield, I think originally noted, there's fractal structure to the lungs in that you'll see the same patterns of the daughter branches, it's asymmetric bifurcation, repeat over and over. We go down through the airway tree. So initially we were utilizing this to come up with a fairly efficient way of modeling the acoustics. More recently, though, then we started getting pretty good CT images. This is a CT image of the airway structure of a pig, and it doesn't really follow this fractal structure. So we were going to kind of more of a brute force analysis of the acoustics through this airway tree. Now, even though it's complex three-dimensionally, in terms of the acoustics, we can still consider this something what we call a deep branching waveguide. In terms of the airway are such that we can really think we have this quasi-planar waves propagating down through the airway structure, and then they continue to branch and branch and branch. So we had adopted this approach. We had done a number of so we built models. We could test our acoustic of this, and what I'm showing here is we would sound into this structure here with this branching airway network. We would use laser vibrometry to measure the surface of vibration on that phantom created by that sound field. What I'm showing down here, I can't remember which is which. I think the right here is the experimental data at a certain frequency. The left is a simulation. It's a mirror image type of phantom. Basically confirm that our acoustics modeling, what modeling of the surrounding solid tissue was accurate. Normally, we've with one of my current Ph.D. students, Brian Henry, we've been studying Professor Omer and Tahe's numerous articles on the airway structure. And really, Brian was looking at using constrained constructive optimization and a few other techniques for airways beyond what we could see on the CT scans. But at this point, we've come to the conclusion that what Professor Tahe has been doing works best for us. And with some help from her, we've been starting to look at that approach in terms of taking the base tree from a medical image and then growing many airways we want to, all the way down to the alveoli if need for a particular simulation. So just a few more images of this. Again, this is the pig lung. And so, of course, within the acoustics, so this is a picture of Brian here, and he's actually here in the room with me. If there's any specific questions about some of this stuff, I might need him to help me answer some of them. So in terms of the acoustics, we can go the way we were doing if we had this field structure where we had this very precise symmetric dichotomy where there's these patterns repeated. It's efficient acoustically that we could calculate acoustic impedance starting at the alveoli and just march up one line all the way up to the trachea using a kind of recursive formula. And then we could, if we have that, then we would have to march down branch by branch to calculate what acoustic field is given acoustic input. And primarily we've focused on acoustic inputs being in the trachea for this initial development of the simulation model. Okay, so that's what some of the equations that relate to calculating those pressures. Now, with these new models that don't precisely follow the fractal structure, we do have to go through every single airway branch and make the acoustic calculations going up the tree to get the acoustic impedance and down to calculate the pressure field. So, the advantage of this, we don't need to have an airway tree that strictly follows the so-called Horsfield model. Disadvantage is it's a bit more computationally expensive, and perhaps 10, 20, 30 years ago might have been unrealistic, but these days it's not a big deal, and Brian has integrated into our programs an algorithm that can actually do this within a matter of seconds for airway trees that have tens of thousands of airway segments. Here are some of these examples of this. Here we're introducing sound at the top of the trachea at a particular frequency, and we're mapping out a prediction of the acoustic field throughout the airways. Here it is, this is like we call a snapshot of the real part of the acoustic pressure. This is a snapshot of the magnitude of the acoustic pressure at a particular frequency. When we calculate the acoustic pressure in the airways, we can calculate things like the velocity of the walls of the airways, and that's important because that is how sound is then escaping the airways and getting into the surrounding parenchymal tissue, and ultimately into the rest of the tissue surrounding the lungs within the chest. Okay, so other parts of putting this whole thing together is we need a source of sound, whether that be breath sounds, or if we're looking at different types of diagnostic approaches, it could be an externally introduced sound through percussion at the sternum, for example, or through sending sound into the airways through the mouth. And all these are done with fairly low level, non-painful, perhaps actually soothing in some cases, I don't know, vibration and sound levels. We take this and we feed it into our models. The airway tree model is purely analytical. It's a very fast algorithm run in MATLAB to calculate the sound that in the vibration of mechanical wave motion through the rest of the torso. We're typically relying on a finite element model. Early work, we were using boundary element models, which were faster, but were fairly limited in the complexity of the structure that we could calculate. Take all that, and from that, we're able to predict the acoustic field within airways. We're able to predict for example, the velocity, the acoustic velocity of the waves that escape into the surrounding parenchymal tissue. We're able to predict what sound field might reach the surface of the chest. So we tried to do some experimental studies to validate this best we could. An example where we had this is an excise preserved lung. I forget the name of the company. And so it's definitely going to be different than in vivo, although it certainly felt like the lung feels in vivo, but certainly there are changes with the preservation process, whatever they used, alter things. But it gave us a nice stable experiment form that we would introduce sound into the lungs. We would use spirometry, that's this grid shown here, to map out the acoustic field that was treated to the surface of the lungs, and then we could try to predict this with our model. So we had our airway tree model. We were predicting the sound field in the airway tree that integrated into our model of the parenchyma, predicting the sound field within the parenchyma. And we can predict what we're seeing on the surface of the lung. And so we were seeing some of the same general features in terms of hot spots, for example, at various frequencies. Although it's not perfect, but we thought it was pretty good. We certainly haven't seen studies that have quite with this level of sophistication spatially in terms of the whole structure. All we've done on pigs have been, here we're percussing auditory stimulation to the sternum, and we're taking measurements on the back of the pig, and introducing a collapsed lung. And the two different pigs, we're showing that over the range, as you move into the higher frequencies, there's fairly consistently a drop in the sound energy level that reaches the back when the lung is collapsed. And basically, we're showing that, yes, we're capturing these trends, even if you look on the same magnitude scales as we were seeing in the experiment. Also, we're doing the sonification studies with these poor pigs, similar types of things. And here, this is the, I think it's the average about a half a dozen pigs, and our simulation is the blue line, all roughly the same size. Here, when a pneumothorax is introduced, maybe we don't see much of a difference here, but if we start to look at the acoustic energy in frequency bands, we can consistently separate the cases relative to once a collapsed lung is introduced, in our simulation here is the blue. Okay, so model, we can start to look at other things that we can't at this point experimentally validate, although I would hope to in the future. We can, in this example, we're sent to the airways and we have tumors introduced, and how the acoustic field is altered by the presence of a tumor. We don't have a good way of being able to experimentally validate this. I do hope we will in the future using hyperpressurized gas magnetic resonance elastography. As it addresses, I think this is something that it will be those types of confirmatory studies. Studies, for example, on fire, where a shear vibration to the side of the lung, and we're showing what effect process of stiffening of the interstitial tissue will do in terms of altering the wavelength, the shear wavelengths. Okay, we've all done some limited human studies on normal subjects looking at percussion. Here we're showing the sound transmitted to the back under normal conditions. Here what happens if we have a collapsed lung on the right and there's a reduction in the sound transmission to the back. We could exactly run this experimentally. However, what we did do is experimental studies on patients undergoing elective surgery, laparoscopic surgery, where a thorax was induced. And we could measurements of how well the sound gets transmitted, and just like in the pig, we see a consistent reduction in the energy level within certain frequency bands that is consistent with our simulations. So kind of at this point, to summarize our progress, we've developed these techniques to understand the pulmonary system acoustics, infectious disease, injury. We've entered a number of modeling innovations in terms of utilizing fractional models of viscoelasticity, plano theory, lungs, this in terms of modeling of the airways. We've had a number of innovations in terms of measurements. I mentioned a few things, different techniques we use, optical or magnetic resonance elastography. And we've been developing these programs. So at this point, I think this is our website. We have these from our link. We've developed these in MATLAB and designed such that you don't need MATLAB to run them. Executable, you do need to download something that's free from MATLAB, but once you have that, you can programs. And so I think my student Brian put together like a 2-minute video. I'm jumping around in our here. And first of all, let me stop it here, and I have to admit that I need to wear glasses to look at my screen here and see the small print. Basically, what we have here is we're starting to build a database of cases that we have run that then this software can be used to visualize. The finite element cases, they take a while to run, so it is something that at this we need to pre-process. Right now, that's also how we're doing the strictly airway acoustic studies, although in the near future, we expect to have that integrated so that it's done on the fly as you're running the executable, because, again, the calculations are relatively fast. Let me see what, in terms of other things, depending on the case, we'll have different look at, different components of the torso we can look at. And so this is an example where we're there's a shear force applied at the side of the torso. We have surface image renderings, as well as we can take slices through the body at different points. And this geometry here is based on the visible human male, showing the magnitude of the, I guess, anterior posterior vibratory motion created by the shear wave force on the right. Again, here we can go in and we can, this is an animation of the anterior motion waves propagating laterally at different frequencies. The kind of a snapshot here where we're focusing on the lungs, and in this case we can compare a case of healthy versus fibrotic at a particular frequency and look at how that has the shear wavelength. Oops, okay. Let's jump here to wherever I was. Okay. And one more, I already showed that one. Okay, one more example. This is one of the cases where we've sent into at the edge, starting at the top of the trachea. On the left is the magnitude of the vibratory motion in the anterior direction on the torso. Here it is, looking at the rib cage. And we zoom in and look at the lungs. On the right is, again, is like a cross-sectional image of taken at a particular height. And we can also, then you can do animations, look at motion in a particular direction, looking at a particular slice, and you can pick the slice to be sagittal, coronal, lateral. Those were a few quick examples of the AD.exe can currently do. I mentioned at the beginning, some of the things I do in the future is take advantage of some expertise we have particularly at UIC in visualization of reality. And actually, I'm going to get here, and actually this is a study we recently did. This is an excised prostate. There's a radical prostatectomy due to cancer suspected, and we would MR elastography on these prostates, and then on them we could reconstruct of the mechanical properties throughout the prostate, and did have that confirmed by pathology, and hand this over to Christian and his group, and be integrated into their augmented reality environment. So again, the two-dimensional screen does not do this justice, but dimensions, and we have a surgeon here, Simone Crivellaro, urological surgeon, has a stylus that he can move around, and then when he goes and starts to poke into the prostate, he will feel how stiff it is, depending on where he is poking. So the idea behind this, in this particular case, is once we do this in vivo, is that this will give the surgeon additional information, better orientation of what it is they're going into when they perform actual surgery. So we're not there yet, but looking into the future, and that's what hope that we also there were different examples within the pulmonary system where this type of rendering, this type of information visualization could be helpful in terms of diagnostics, in terms of planning, treatment planning, as well as certainly being an educational tool. And a lot of what Christian does is focus on education, on being able to simulate different types of procedures. That, I have some music in case you fell asleep. This simulation done by one of my former Ph.D. students, Kaya Gasser, and I'm putting in a plug for his new startup company here. He is involved in continuing to make these types of simulations for different medical applications. There it is, Spherical Chicken Studios. I hope that's okay by NIH. Guys, that I could plug my former student's company. It's this is here. But with that, that's the end of my formal talk, and I just, again, thanks for your attention. I'm acknowledging here the names and photos, some of my colleagues, Richard, top left, Dr. Bob Bach, bottom left, and a bunch of my students, some that have been indicated before, Dieter Klatt, lower right, my current collaborator in MR elastography. Certainly, thanks for your attention, and happy to take any questions.
Thank you so much, Tom. I'll open up for questions, and please, everyone, please unmute your mic and unmute your webcam so we can see your faces. Any questions?
Even though you show some validation slides, it seems to me the accuracy of your model and particularly local alterations of the lung disease. Can you comment on that?
Yeah, no, that's a good question. I think, yeah. So, well, we've done a lot with pneumothorax. So, but that's a larger thing. We're very confident about that. Last year we've been focusing more and more on the airways and to look at various diseases of the airways that are going to occur, for example, down the alveolar structure and in the bronchioles. And no, we need to do more work in terms of experimentally validating that. I think we have experimentally validated fairly well the acoustics of the airways and our ability to simulate how sound travels through this branching network. But we haven't not yet looked at simulating specifically certain diseases of the airways and exactly what effect they will have on the acoustics. We can certainly speculate, but we haven't done that, the experimental validation of that yet.
So, Ching, since you brought up the first question, I was curious whether or not you and Tom think that there's any way your models could be used maybe on this project in terms of your airway model.
Certainly, because we have a model that can create subject-specific airway model from trachea down to the 20-some generations of the airways. And then for like male, female, COPD, asthmatic subjects. So if, say, Tom would like to use these models to this acoustic model, he is more than welcome to use them. Another question for you. You showed the acoustic ratio in the airway tree. Did you rigid airway or do you assume breathe-in airway, like human airway model?
Okay, well, to answer your question first, yeah, these are compliant. These are deformable. We are modeling the mechanical structure of both the cartilage as well as the soft tissue and looking at the cartilage fraction as we go down through the airway. So that is all taken into consideration, and we need to have that because we are predicting how much the airway walls are vibrating, and then that is our input to predict how much sound is then traveling into the surrounding parenchyma. And to also comment on Grace's question, yeah, I thank you, Dr. Lin. Yeah, we think to some degree we may be using some of the same modeling now that we really have adopted a lot of what Dr. Tahe is doing in terms of her algorithmic development structure. Although I know you guys really some cool stuff with the imaging combined with that and looking at how the airway tree changes throughout the respiratory cycle. And yeah, I very much would look forward to communicating further and seeing how we can utilize what you're doing within the sort of simulations we're making of the acoustics. Thank you.
Mm-hmm. Sorry. you consider turbulent flow in trachea in your model?
We have not done turbulent flow in the model. We haven't. We've been focusing more on the acoustics. In previous work, I've looked at turbulence and transitional flow in the vascular systems, and that was actually with Paul Fisher, who's now at UC. And I have seen some of your recent work looking at simulating the turbulent field. I think that would be the input in terms of then giving us the spatial and spectral structure of the acoustic disturbance that then we would input into our model to predict how that acoustic disturbance will propagate through the airway tree and the surrounding structure.
In terms of your do you have a public interface that people can use for the Audible Human Project at this point?
Yes, we do. And now you guys can see my whole screen here, right?
Yeah, I was wondering if you can do a little demo from.
At the risk of on the fly here, let me just open up a new window here. And sadly, I'm going to go roundabout way to get to my website here. Here's the actual website. So you can Google Audible Human Project, Royston, you'll find this. At least for me. And then it's under these downloads, you to this HP visualization interface. And we do have tutorials here, which we're constantly trying to improve. Go here and download the latest version. And you also can download that MATLAB, that free MATLAB thing. There's a link there. MATLAB runtime library. And then basically what you will have is give you these files, which you can extract in so I'm just running the executable and hoping that it works. Yes, okay. And we need to, we're acknowledging my student, Brian, and I think we need to acknowledge NIH on this page here as well. And here it is. And so here's the kind of the current base of files, and this is with the little session we had before, where you can pick one of these, and I don't think we looked at these. Depending on which one you pick, you have different parameters then on the right. Things we can look at, and different attributes we can adjust. Okay, where am I, Brian?
It takes a few minutes.
Okay, I picked the big comprehensive file that takes a few minutes to load. Okay, and you can adjust the orientation, rotate it. You can adjust the color scale. Gives a little bit of information. Here, I want to not hang upside down. I'm going to take it. Yeah, okay, there we go. And
people that you can see a bigger version if you double click on the shared portion of the screen if you have trouble viewing.
On my laptop here, it's a little. I did pick one of the more complex cases here. The visual rendering is going a little bit slow. But I don't know. Is that it's there to play around with, and we are constantly making updates to it. It's a database of different cases that one can explore in the near, at least for the airways. We hope to give a little more capability in terms of being able to look at the cases that are here already simulated, but that you could simulate on the fly. Okay.
Yeah.
Okay. All right. This is so that to download it.
Are there questions for Tom? I want to give you our virtual applause. Thank you very much.
Thank you very much. Thank you.
And will be posted on the IMAG wiki under your webinar. And thank you again for everyone for participating on this webinar.
Thank you.
Thank you. Thank you.